So, hello everybody. It's my pleasure to be here today, so I thank the organizers for inviting me. And it's a pleasure also to present the work we are doing on Xylella fastidiosa outbreaks in France. So, no. Yep, okay. So, as Sandy already said, Xylella recently emerged in Asia and in Europe. It was detected in 2012 in Taiwan on grapevine, then in Italy in 2013 on Italy, and Sandy shows a lot of pictures. In 2014, Iran reported the presence of Xylella on grapevine and almond trees also, and it was the turn of France to declare Xylella on various ornamentals in 2015, and last year, Germany declared a case of Xylella in a greenhouse on ornamentals, and later on, during the fall, Spain reported the presence of Xylella in Balearic Islands on a wide diversity of plant hosts and with various strains. It should also be mentioned that from 2012, various interceptions of contaminated coffee plants were made throughout Europe and involved several strains of Xylella. So, and this emergence of Xylella in Europe is a burning issue, as attested by these covers of magazines and newspapers. In France, since the first case in natural setting has been reported in July 2015, more than 18,000 samples have been tested for Xylella, and this sampling effort is of course very important in the contaminated or nearby area of southern France, including Corsica. But more generally, the southern half of France has been widely tested for the presence of Xylella. And so far, the infections are mostly located in Corsica, here, and in southeastern France. A few contaminated spots have been declared in Île-de-France, near Paris, and in Pays de la Loire, but they concern mostly interceptions of coffee plant material. In Corsica, 340 foci have been so far detected, and they involved 34 plant species. And in contrast to what we saw before, not all foci include Polygala myrtifolia. So there should have been other introduction of Xylella done by the mean of contaminated Polygala myrtifolia plants, because 20% of the foci do not involve any Polygala myrtifolia plant. And it should be mentioned also that 6 foci have been detected at high altitude near 100 meters. So it's quite high, and the conditions were not supposed to be very prone to bacterial multiplication. In southeastern France, in the French Riviera, 20 foci have been detected, and they involved 5 plant species so far. And here is the list of the 34 plant species that have been detected contaminated by Xylella, so have been declared host of Xylella. As soon as one plant is detected contaminated by Xylella, it is reported as a host in France. Okay? So we can discuss that. And the 6 most frequently detected contaminated plants are Calicotum villosa, Polygala myrtifolia, Spartium gynceum, Helichrysum italicum, Cistus montpelliensis, and Lavandula augustifolia. Most of these plants are indigenous to or are common to Mediterranean and Corsican scrubs. And at least 2 of them, the curry plant, Helichrysum italicum, and the lavender, have a regional economic impact as they are used to produce essential oils used in perfumery. All other plants are mostly ornamentals only. And these 5 plant species, olive tree, strawberry tree, citrus, eucalyptus, and grapevine, while being present and largely disseminated in the contaminated areas of Corsica and southeastern France, have so far never been detected contaminated by Xylella. And the sampling effort is quite large, especially on olive trees. Okay. The plant materials, one sample, is analyzed by the qPCR test proposed by Harper and colleague, which target the RIM-M gene, which is a 16S RNA processing gene. And this test is run directly on DNA extracted from plant material. There is no compulsory isolation of strain in France. And then, for the positive or undetermined samples, then the identification is made based on the amplification of 7 housekeeping genes. And once again, this typing is made directly from plant material. So just a quick reminder about MLST and MLSA. These are widely used methods for the taxonomic assignation and the typing of strains. They are based on the sequencing of the partial sequences of 7 housekeeping genes, genes that are chosen because they evolve slowly and they are under neutral selection. Here is the list of the 7 genes that have been proposed in the MLST scheme for Xylella that was proposed more than 10 years ago. And a set of 4,000 sites are under analysis. And so far, they ended in the description of 80 sequence types for Xylella fastidiosa. So it's a quite genetically diversified species. And in the frame of the control plan in France, the importation of coffee plants is subjected to analysis. So far, 21 plants were detected positive out of 135 plants that were tested. So this represents 14 interceptions of contaminated plant material. When the origin of these coffee plants is known, they come from Latin American countries. And it should be mentioned that some of these countries, like Ecuador, never reported the presence of Xylella before. The genetic analysis of the strains that were isolated from this plant material reveal novel alleles and novel sequence types here that are in red. And some of these sequence types that are underlined here were detected not only in France, but also in Italy and in Netherlands. So this means, as Sandy said, that the importation of ornamentals, and among which coffee plants, represents a high risk of introduction of Xylella fastidiosa and genetic diversity of Xylella fastidiosa to Europe. So let's focus now on the outbreaks in natural settings in France. So in 2015, 431 samples were typed using the MLST methods among the 531 samples that were declared contaminated by Xylella based on the qPCR test. And among the typed samples, 25% were not completely typed because at least one housekeeping gene was not correctly amplified or sequenced. In 2016, 321 samples were typed among the 332 that were declared contaminated by Xylella, and the percent of incomplete typing was higher, 45%. So there is ample place for improving the direct typing of Xylella from plant material. And we should take into account that there are a lot of PCR inhibitors in the plant macerate, and we have to deal with a large range of plants. And we should also consider improving the protocol by proposing various annealing temperature or specific annealing temperature for each gene. Well, there are other proposals that can be made. Various teams are working on that also. So here is the phylogeny of Xylella fastidiosa based on well-known strains and some French isolates. So here in red, you have the multiplex subspecies with the type strain here, then the most subspecies, which is linked to the multiplex subspecies. Here you have a clade which group the fastidiosa subspecies and a group of strains that were sometimes assigned to sandy subspecies that were mostly isolated from coffee plants, and the real sandy subspecies with the N1 strain, and here the more divergent Poca subspecies. And most of the French isolate fits in the multiplex subspecies as indicated by the grouping of these samples close to the multiplex type strain. And these samples were contaminated by two different lineages that are well known in the States as ST7 as ST6. ST7 strains were so far isolated from almond tree, red oak, olive tree, and black sage, while the ST6 strains were so far reported from almond tree. Besides ST6 and ST7, we also identified one recombining ST. As you can see here, this ST79, is made of one allele of cis-G gene that is common in the sandy ST76 scheme. And the other allele of this recombinant ST are typical of ST6 and ST7 here of multiplex. So this recombining ST should have generated by strains from the multiplex and the sandy subspecies. We also identified one focus in Corsica that was contaminated by a more classical sandy ST76 that was also reported from coffee plant. And more importantly, one focus of Polygala myrtifolia at the Italian border in Menton was reported contaminated by the ST53, the one that contaminates olive trees in Italy. And 2% of our samples are considered as undetermined. What does that mean? This means that for this sample, the genetic analysis of the 7 genes is not strictly conclusive. While the DNA chromatogram, as you can see here, are generally of very good quality, at some punctual positions, there are two overlapping nucleotides that we couldn't discriminate. And these positions are usually very important to discriminate among alleles of the genes. So in these cases, we couldn't properly assign the allele because of these superimposed peaks. So for these 2% of samples, we have some alleles that are properly assigned, and for some genes, some alleles couldn't be properly assigned. Okay? And this certainly is the result of the presence of several strains or several recombinants in the same plant, and which should also indicate that isolation of different strains from the same plant has been already made. So it's a reality. So far, what we found with this genetic analysis of the strains in France is that we have diversified the Xylella strain in France, which could indicate that we faced several introductions in the past and that maybe some diversity was generated locally via recombination. So far, we couldn't see any host specificity of the multiplex lineages, and we couldn't also apparently see any specificity in the spatial distribution in Corsica or in southeastern France of the multiplex lineages. And the direct typing of Xylella from the plant material shall be improved, but yet it allows the identification of up to 75% of the samples, and only 2% of the sample present mixed infection. The 3 first strains that were isolated on media were sequenced. These strains were isolated from Polygala myrtifolia and Spartium janseum, and they were sequenced using the Illumina technology. And I will present today the comparisons that were made using the CGView comparison tool. With this tool, the genomes are virtually cut in 1 kb fragment, and these fragments are blasted against a reference genome. The results of these blast-end comparisons are shown in map here, where each circle represents a genome involved in the comparison. And the color is used to indicate the percent identity of the blast hits, and the darker the color is, the higher the identity is. So if we compare the 3 French strains, what we see here is that first we took one of the ST6 strains that was isolated from Spartium gynceum as a reference strain, and we compared it with the other ST6 strain. It's represented here in the external circle. And we also compared it with the ST7 strain isolated from Polygala myrtifolia. And the result of the comparison is represented in the internal circle. So what we see is that the 2 ST6 strains are mostly identical. And the ST7 strain, in contrast, is a little bit more divergent with all these red and white stripes dispersed all along the genome. And what is interesting here is to see all these white stripes that indicate that a fragment, a DNA fragment, is absent from the ST7 genome sequence, but is present in the ST6 sequences. And these fragments, in fact, correspond to a phage sequence, an entire phage sequence, that is present in the ST6 genome sequences, but is absent from this ST7 genome sequence. And if we make this kind of comparison with the closest US relatives of our strains, we get this map. So here we use the Dixon strain, which is a ST6 strain isolated from almond tree as a reference, and we compared it with the 3 French strains, and we also involved 2 ST7 strains from the US in the comparison. And what we see, apart that strains are more similar within an ST than between the ST, is that several fragments here that are grouped on the genome sequences are absent from all strains except the Dixon strain. And these fragments, in fact, correspond to one plasmid that is present in the Dixon strain and that is known to harbor an entire type 4 secretion system. And this system is known to be involved in DNA mobilization and DNA conjugation. So this means that the strains that are devoid of this plasmid could maybe be less prone to recombination than the other strain. So this could be a good news for French strain. We also use these genome sequences to infer divergence time within the multiplex subspecies. This analysis involving root-to-tip regression was done in very close collaboration with Adrien Rieu from le CIRA de la Réunion. And here you have preliminary results because we do not have access so far to enough genome sequences. But the results are quite interesting and are worth mentioning. So here on this dendrogram, what you see is that this ST7 strain, isolated from Polygala myrtifolia, should have diverged from its last common ancestor shared with its closest US relatives, M12 and Griffin, around 49 years ago. And here you have the confidence interval of the time of divergence. It's quite high. And similarly, the 2 ST6 strains isolated from Spartium gynceum should have diverged from their closest US relatives, the Dixon strain, around 35 years ago. And here is the confidence interval. That means that we may have faced introduction of Xylella far earlier than what we thought. We were thinking about 15 years ago, but it's maybe more 35 to 50 years ago. So we fulfilled the core postulates for one ST6 and one ST7 strain on Polygala myrtifolia. And here you can see that 13 weeks post-inoculation, we observed these quite typical symptoms on twigs of Polygala myrtifolia. The strains was systemic in the plant, and we could re-isolate one strain from these symptomatic plants. So, is there any insect that can be considered as a vector of Xylella in France? So far, 10 species that are known anywhere else to be able to transmit Xylella fastidiosa were sampled in Corsica. These 10 species are listed here. And among these 10 species, there is the meadow spittlebug, Philinus spumarius. So far, 330 samples of Philinus spumarius were analyzed for presence of Xylella fastidiosa. These Philinus spumarius were sampled in Corsica in 2015, and around 8.5% of them were declared positives. Only 2 other species, Lepironia coleoptrata and Cicadelidae viridis, were also tested for the presence of Xylella and were tested negative. So this is not proof, an evidence that Philinus spumarius can transmit Xylella in Corsica, but it's the first element. So, as you all know, pathogenicity tests involving Xylella are quite difficult to conduct because they should be conducted in confined conditions, and we have to wait for a long time to get symptoms. So we wanted to see if there is any possibility to predict the host range of a strain from its genome sequence. So in fact, this is similar to identify sequences that can be specific to a group of organisms that are expressing a given trait of interest, here the pathogenicity on a given host range. Several tools already exist, but it's not easy to obtain an exhaustive list of targets. So one bioinformatician colleague of mine developed another tool, SCIF, for specific chimer identification. And this tool is able to identify both SNPs and genes that are specific to a group of organisms that are defined by the user. So here is the workflow. It's quite simple. You have to divide your strains in 2 groups. One group with the trait of interest and the other group without the trait of interest. So here, the pathogenicity on coffee plant, for example. And then you upload the genome sequences of these 2 types of strains. Each sequence is cut in K-mers. K is usually 22 nucleotides. It's apparently the best solution. And then an abundance matrix of all K-mers are constructed. And if K-mers are overlapping in one genome sequence, then they are concatenated into long mers. And these tools, finally, allow to have a list of K-mers that are specifically present or specifically absent in the group with a trait of interest, a list of long mers that are specifically present in the group of interest, and the coordinates, of course, of these K-mers and long mers. And here are the results if we do that on a set of strains that are infecting coffee and that are genetically divergent, as it is the case for coffee-infecting strains belonging to the Fastidiosa sandi subspecies, but also to the Poca subspecies. And if we do that, we find 11 candidate proteins that are sequence specific to these coffee-infecting strains. And what is interesting is to see that 5 of these candidate proteins present non-synonymous signature. So this means that they could have faced adaptation to coffee. So now we need, of course, functional analysis to confirm this result. So I want to thank all the persons who participate in this work, and especially Nicolas de Nancy, who was a postdoc in my team for 2 years. I want to thank also the funding agency, la Région Pays de la Loire, for its support, INRA for its support, and of course the European project Ponte. And I also want to thank my colleagues from ANSES, who largely participate in this work, and especially Bruno Legendre. I also thank Maria Saponari for sharing the strains. And I thank you for your attention.